Hello, everyone. Welcome to another episode of Q&A about history of science and technology. This is actually my third live stream of today, and there'll be even one more after this as part of my day job, designing the new version of Wolfram language. But this is a break, so let's see. Susie asks, do you know anything about the history of vaccines? When was the first vaccine developed? For what? I don't know this in as much detail as I probably should. I think Edward Jenner, Louis Pasteur, those are two famous names from around 1800 and mid-1800s. The kind of the first story had to do with smallpox, and the idea. So I'm not sure who first had the idea that you could kind of give a partial. Well, okay, let's start from the beginning. People have known for a long time that if you get a particular disease, you'll probably only have it once. You know, if you have chicken pox, chances are you're only going to get chicken pox once in your life. So then the question, I guess, was, well, can you sort of give people some imitation of the disease that gets whatever it is that makes them not have it again in their lives be a thing that works without having them get really sick from that disease? And that was a thing around 1800, this idea of cowpox versus smallpox. And could you give an infection with something which was kind of like the thing that was the bad disease, but have it not be the bad version of the disease? And I think that was something that people sort of empirically, that was kind of the deduction, give a less bad version of something that's like the disease to make one immune in the same way that having the disease would have made one immune. And I think then the kind of development of other vaccines probably waited for a better understanding of the immune system. I mean, as I think I've talked about in other cases, sort of in humans, there are two basic pieces of the immune system: the innate immune system, which just kind of tries to notice things which are kind of alien substances, alien infective agents, and so on, tries to notice them by sort of general patterns of, oh, if the molecules look roughly like this, they're probably bad. That's kind of the innate immune system. Then there's the adaptive immune system, which is kind of an invention of the higher animals like us, which is something where you are actually producing antibodies and T cells and all those kinds of things, where you've specifically, there are a large number, maybe 10 million, 10 billion, maybe a trillion different possible antibody configurations, which are all molecules of different shapes. And those molecules of different shapes, if they match with a bad molecule that's coming in, then you amplify the number of molecules of the shape that matches the bad one. And then there's a whole cascade of activity of the immune system that comes from that. And there's a lot of trickiness associated with when you're young. The immune system has to kind of learn what are molecular shapes that are ones for you versus ones for the alien outside world, and not react to the ones that are for you. If it does, you get autoimmune diseases and so on, but react to ones that are kind of alien and from the outside world. But that whole theory of kind of the sort of the natural selection theory of the immune system, the idea of sort of immune. Well, okay, so the kind of what the stuff of the immune system is, immunoglobulins, antibodies, things like that, that was figured out, I think, around 1900. And just sort of the nature of the kinds of things that are in the immune system, and the different kinds of immunoglobulins, the different and so on. That was figured out as a sort of biochemical matter, I think, around 1900. And then it was probably the 1940s, 1930s, 1940s, the clonal selection theory of the immune system, which was this theory that the immune system kind of has all these possible antibodies produced, and depending on what antigen is seen, will amplify the antibodies that match that antigen and so on, and the corresponding thing about the sort of avoiding sort of problems with things that attack the self and so on. That was figured out, I think, in the 1940s, if I'm not mistaken. Burnett, I think, was the name of the chap, along with oh my gosh, Nils Yerner was another person much involved in that. Anyway, then I think that there were various sort of big efforts to do immunization, of which one of the very famous ones was polio. And there was sort of a question of, once one had some understanding how the immune system worked, then the issue was, well, okay, how do we give something which is kind of like the antigen we're trying to get the immune system to respond to and generate antibodies to, but not something which can actually make people sick? And so there were a couple of different approaches. There was the idea of kind of killed virus, which is the thing is not alive, but it's got the same kind of shape. There's sort of attenuated virus, where the virus has had things done to it that prevent it from being able to, I guess, replicate and generally be as aggressive as it would normally be. But one of the sort of key kind of cases of this was polio, and that was what, in the 1950s, I guess. There were big efforts to try out different vaccines. There was, I think, the Sabin vaccine and the Salk vaccine. And they had different approaches. One was a killed virus, one was a attenuated virus. I'm not sure. Don't remember exactly how it worked. But there was then something which was kind of a new idea, a huge public health trial of, do these vaccines work? I have to say the only part of this, I don't know this history terribly well, but I did know Jonas Salk to some extent later in his life. I think there had been a big fundraising effort for sort of a cure polio effort, I think from maybe the March of Dimes charity, I'm not sure. But anyway, when after this big public health trial, people sort of got convinced that yes, one could vaccinate against polio, and that actually worked. But then it's like, but we raised all this money to cure polio, and then what ended up happening, I guess, was that sort of money was essentially given to Jonas Salk to do something good for the future of science, and he built the Salk Institute in San Diego as kind of a thing to sort of develop future things in science. And Jonas Salk himself became interested in, well, lots of, I would say, more philosophical kinds of issues. And as I say, I'm in the, what was it? Must have been the 1980s. I did know Jonas Salk a bit. I think the polio vaccine was, I think, one of the sort of big success stories. Then by the 1960s, when I was a kid, at least in England, there was starting to be more and more vaccines. There were vaccines for mumps. No, actually, that was later. Measles was one of the ones, because I know I had mumps. So there wasn't a vaccine for mumps in the early 1960s, at least being given in England, nor for German measles, chickenpox, these kinds of things. Those are all vaccines that became kind of popular I don't know whether they've been developed at all, but they certainly became popular probably by sometime in the mid to late 1970s, maybe. Sort of the tricky thing with the vaccine is, first you have to figure out, well, you take the critter that is going to attack you, then you have to find some way to get a piece of the critter that's going to be sort of a characteristic piece. Like it could be the capsid that's kind of a head of the virus. It could be some other piece of it. You have to figure out how you're going to sort of that. Then you have to figure out that's kind of step one, is sort of what is the thing you're going to expose the person to to get their immune system to respond as it would respond and prime itself for when that actual attacker comes. But the second problem is that a vaccine is something, you just inject it into some muscle somewhere. It's got to be the case that the immune system mounts enough of a response to that it's actually significant. Because otherwise it's like, oh, there's a little tiny bit of fluid in this muscle, and who cares? The immune system will do some local attack, but it won't actually sort of generate all these antibodies that could potentially last a lifetime to protect you against that disease. And so the kind of trick that's used is this notion of an adjuvant, which is some substance that is going to sort of get the immune system all riled up, that gets injected along with the actual sort of killed virus or whatever it is. And it's kind of a black magic story of adjuvants and what kind of thing might be in that adjuvant. Is it? I think there are some that are based on killed bacteria, I think, and there are some that are based on have some mercury-like stuff in them, or I forget all the details. But some of both a lot of kind of reactions to vaccines and the effectiveness of vaccines depends on that whole adjuvant story. And I think that was, I don't know exactly when that was worked out. I think the adjuvants that are even used today are quite old. They're things that have been used for a long time. I don't think there's so much development in that area. So then, vaccines became more popular. The things like flu vaccines, flu vaccines are somewhat unheard of in some countries. In the US, they became popular. I don't know, I started getting them 20 years ago, maybe. That's always a challenge because the flu virus evolves quite a bit over the year, and so you have to kind of make some level of prediction of what will the next season's flu be like to make a vaccine that's actually going to be effective against it, and it doesn't always do that well at doing that. I mean, there are many mysteries in infectious diseases and immunology and so on, like the seasonal dependence of flu is something still not well understood scientifically. I mean, there are theories about it, oh, people are inside more, oh, your sort of nasal cavities get dried out if it's cold, and so that makes the innate immune system not work as well. And there are various theories, but I don't think there's a well-established understanding of why there is that seasonal variation. So there's sort of very basic things that are not understood. Now, in more recent times, kind of there's sort of a general view that one is just doing by giving vaccines, one is just doing the same kind of thing that one does if one had gotten chickenpox and gotten immunity from having had chickenpox. It isn't really true. There are a lot of detailed differences. Whether those differences matter is a complicated question. That's a whole immunology story, which maybe we'll talk about some other time. But we're talking about history today, so let's not go down that particular rabbit hole. But then there's the question of sort of the more modern vaccines, like the sort of rapidly developed vaccine for COVID. That was the result of a long-running effort to, well, I should explain something else, which is generally the immune system mounts a defense, an attack, whatever, against foreign things that are attacking you, like some virus or something like that. One of the things that can happen is if you get a tumor is kind of a modified version of your own cells, modified by sort of activating different elements of the genome or whatever else, and having unbounded growth and all sorts of bad things. But it's a thing where it's kind of self cells that are and self proteins that are being produced there. So the immune system isn't as well able to mount an attack against that, because the immune system learnt in your early life to not attack self things because you don't want to attack self things, if you did attack self things, you would get autoimmune diseases. So the immune system tends not to go after tumors. Now, it probably does go after them a bit, and maybe all the time we're generating small tumors that are being destroyed by our immune system. That's been a long time theory. But then the idea, which has existed for probably 100 years, is, well, could we somehow get the immune system to start going after tumors? Could we do kind of an immunotherapy where we somehow get the immune system to say, oh, yes, I know this tumor thing is something bad, let me go after it. So people had tried to do this for a long, long, long time, and it's sort of not really worked. And it's been kind of a complicated story. It's been one of these things which sort of should work, but the devil is in the details, and the details didn't work out. But then there were a couple of companies which were starting to try and develop cancer vaccines based on mRNA, and based on kind of just going in and sort of having the thing that you were supposed to be getting immunized against be something that is just being generated within your cells and so on, and have that as a way to kind of expose you to that antigen. And so there was this effort to make cancer vaccines. They had gotten a certain distance, but then when the COVID virus came along, it was like, well, that technology can be used to make a vaccine very quickly, just knowing the sequence without having to mess around with all sorts of details about viruses and this and that and the other. I mean, I should explain that another approach, and it was an approach used for some of the COVID vaccines, was to use a adenovirus, which is a virus that will go in and infect cells and take with it, and it's just a particular kind of virus that you can put another sequence into, and when it goes and infects a cell, it will pull along with it the sequence that you want to immunize against, so to speak. But in any case, that was kind of that was sort of a retargeting of what had been intended as a cancer vaccine. And there's a lot of technical detail about making sort of these microscopic cell-like lipid nanoparticles to be able to deliver, to be able to have these pieces of mRNA actually get inside cells and so on. Maybe it's interesting to talk a little bit about the history of DNA and RNA and all those kinds of things. The molecule, okay, so back in the 1800s, people like Mendel had noticed these sort of rules of genetics. He was pretty lucky with these pea plants that he looked at, that they were sort of simple enough in their genetics. You could kind of trace what was happening. It's a little bit like human eye color is another sort of simple enough piece of genetics. You can kind of trace, brown eyes plus blue eyes, parent with brown eyes, parent with blue eyes, typically gets child with brown eyes because blue is a recessive trait. I always found it very confusing, all of the kind of notation and things about recessive traits and so on, and these what they call Punnett squares, which are the kind of what happens if you cross sort of a parent with one characteristic with a parent with another characteristic and so on. It's a very sort of it's not explaining why it happens. It's just sort of explaining some sort of essentially combinatorics of what goes on. That stuff was known. I mean, Mendel's work was not well known, and eventually was sort of rediscovered, I think around the beginning of the 20th century, I think, well, maybe a tiny bit earlier than that. But this whole idea of kind of genetics by just sort of knowing you have one organism with this characteristic, another with that characteristic, they mate what comes out. It was, I'm trying to remember this history. I think it was the 1920s, 1930s, a chap called Thomas Hunt Morgan in the US, who did a lot of stuff on fruit fly genetics. But anyway, there was more and more that was discovered about sort of the combinatorics of genetics. Meanwhile, the molecule, DNA, was identified, and it was known that it had something to do with genetics. Now, it had been the case for gazillions of years that people had talked about, bloodlines a lot, and would this particular and sort of the notion was that somehow it was the mixing of blood that was the thing that led sort of the children was sort of the mixed blood of the parents or something, and that blood was somehow the thing that was the carrier of this genetic information. I think it got somewhat more clarified. There were particular molecules that were involved and so on, but the how did that work wasn't known. I think there had been sort of an idea that it could really be something combinatorial, where there's some kind of way of specifying some sort of essentially digital information. I think if I remember right, well, there's a physicist named George Gamow who was involved with that, but maybe later than 1953, because 1953 was the time when Francis Crick and Jim Watson discovered the structure of DNA. What happened, I think, oh my gosh, what's her first name? Franklin, it's her last name. I forget her first name. A woman in London who had gotten DNA as a molecule and managed to crystallize DNA. DNA is a big, long, complicated molecule. She managed to find a way to make crystals of it. Now, meanwhile, in the 1920s, this idea of X-ray crystallography had originated due to the Braggs, the father and son kind of team, William and Lawrence Bragg in Cambridge, had figured out that you could shine X-rays on a crystal and you would end up getting these diffraction peaks, these particular angles at which the X-rays were reflected. So normally, if you have a mirror or something and you shine light at any angle, the light will be reflected back at the corresponding angle. But in the case of when the wavelength gets to be comparable to the distance between the atoms in the material, and if the atoms are arranged in a regular way, as they are in a crystal where it's just a periodic array of atoms, then the effect of the sort of interference effect, diffraction effect, causes it to be the case that the X-rays are only reflected if they're at a particular angle. It's only at that angle that the sort of X-rays that are produced by all the different atoms in the crystal kind of line up, the waves all line up, and you get a diffraction peak associated with reflection at that angle. And so that allows one, that diffraction peak, that allows one to get, well, what is mathematically a Fourier transform of the structure of the crystal. So if you have a crystal with a certain spacing of atoms, you'll get a peak at a particular angle. As you change, as you have, like, let's say you have an arrangement of atoms that's a bit more complicated than just being regular array, but something where there are multiple atoms in different places in like the unit cell of the crystal, you'll end up getting this sort of interspersed collection of different kinds of peaks in this diffraction pattern. And a lot of that was known, had been developed in the 1920s and so on. Lots of materials have been studied that way. It was known kind of what the crystal structures of materials were based on X-ray diffraction. Well, okay, so then the question was, okay, I can't believe I've forgotten this person's name, but Franklin is the last name, who unfortunately died of cancer right around the time all this stuff was happening. But she had produced these crystals. And then in Cambridge, Francis Crick and Jim Watson did X-ray diffraction. And maybe this Franklin person had done some X-ray diffraction as well. But the question was, when you see all these peaks in X-ray diffraction, the issue is, well, what actually is the structure of the molecule, the structure of the crystal that that makes these makes these peaks? Rosalind Franklin, that was her name. Sorry about that. My memory is not too bad, but not perfect. Anyway, the question was, you see these peaks, but the issue is, well, um, uh, what molecule, what shape of molecule will lead to those peaks? And it's not unique. There can be many different shapes of molecules that lead to the same peaks. Well, the big thing that Crick and Watson figured out was, well, actually, the structure that can lead to the particular set of peaks that are observed is this double helix structure. And they figured out this idea that you could have these two strands. I think that idea had been suggested by other people. I vaguely think that George Gamow was involved in that. But I think that idea had been suggested, but they kind of put the two things together. And actually, their paper doesn't really make a lot of sort of noise about kind of this whole digital structure and sort of identical sort of complementary strands and so on. I think it just mentions at the end, oh, this could be what this structure means happens. But anyway, so 1953, structure of DNA is figured out. Then kind of the issue is, well, how does it then work? What actually happens in a cell? How is this DNA used? And that then led over the course of a number of years, I think this was all happening in the 1950s, to the transfer RNA and messenger RNA and this whole mechanism of how proteins get produced from genes associated with DNA. This whole idea of what is a gene was quite muddy. And it's kind of, you've got this DNA, it's this long molecule, it's like some pieces of the molecule are transcribed into proteins, but just how that works was still muddy. It wasn't until the 70s that people really started systematically knowing, oh, there's this sequence on the DNA, and that means that there's this this structure in a protein. I think this whole approach of doing X-ray crystallography for potentially proteins was something that got developed 1950s, 1960s. People were sort of figuring out the structure of myoglobin, the structure of this protein, that protein. There were lots of kinds of big sort of there's a big sort of prize moment when somebody figured out the structure of some complicated protein, because that was difficult to do. You had to kind of infer it based on this carefully done X-ray crystallography. In any case, in the 1950s, kind of the mechanism of how you go from DNA to actual proteins was getting figured out. I guess a bunch of people, I mean, we're starting to get into the domain of people that I met or knew. Certainly Francis Crick and Jim Watson, both people that I certainly met. I guess I knew Francis Crick a bit better than I've known Jim Watson. Francis Crick was when I knew him later in his life, he was always a maker of very wild theories. I mean, he really went after kind of the whole genetics thing. He ended up getting very much involved in the brain. And actually, he went to work at the Salk Institute, which I mentioned earlier. The time when I met him, he was really thinking about, how does the brain work? He had all kinds of theories for what dreams were. He was really trying to sort of have a second act of figuring out brains. And he often had very, very wild theories about things. I mean, Jim Watson has been got more into the kind of policy of things and got involved in I guess, was he director of NIH at one point, things like that, into sort of policy kinds of things. I'm afraid I think like quite a lot of scientists, he was a bit naive about how that would all work. And that kind of came back to bite him in a variety of ways. I think, some not very politic statements that I'm not a big believer in the tread incredibly lightly on things that where you can just say sort of something that's obviously correct, but I think in his case he kind of chose to get involved in issues where there isn't really any sort of clear scientific answer, and sort of just got people upset by doing that. But in any case, And I think the structure of various of these proteins, I remember when I was a kid in elementary school, one of the alumni of my elementary school was a chap called Kendrew, who I think had figured out the structure of myoglobin. And he'd come and visit uh, the the kids, although just seemed like an old chap, and I didn't really know much about what to make of that at that time. But anyway, going into the 1950s and so on, and the figuring out of kind of the mechanism of DNA and such like, a chap called Sidney Brenner, who I knew reasonably well, was the person who figured out about mRNA and messenger RNA and figured out the mechanism by which that works. But this whole sort of the whole progress of thinking about sort of the whole mechanism of proteins and DNA and all those kinds of things, and genetics has been a long-running thing. There are many, many different things to discover. I think when I was a kid, one would memorize the different organelles and cells. And I remember that both the Golgi apparatus and the endoplasmic reticulum were things where the memorized statement about them was they are organelles of unknown function. Well, their function is now well known, and they both have to do with protein synthesis in cells and going from DNA and sort of figuring out and actually being able to construct proteins and so on. I should say that for a while, it was sort of almost a philosophical question. What was a gene? Was it the kind of thing about which you could do Mendelian genetics? Was it something that related to sort of the biochemistry of proteins and things? I think that didn't really get too well resolved until at least the 70s or the 80s. And then in the 70s, it started to be possible to do sequencing of DNA. And there started to be things like the GenBank database, which became NCBI, database of gene sequences. I think I knew some of the people who were involved in that at Los Alamos, actually. A chap, George Bell, Walter Goad, I think those were two names, people I knew, who were involved in sort of computerizing kind of the keeping of data associated with nucleic acid sequences, as they would refer to them then. I think one of the interesting dynamics was, how do you get a community that's doing sort of early sequencing? How do you get that community to actually contribute their results to a centralized database? In many fields of science, that's been a big challenge. People say, I've got my data and I really want to hang on to my data and just use it myself. And the thing I think that happened was, and this was probably when was it? Probably late 70s. The Journal of Nucleic Acid Research just was kind of run by some of these people, kind of put its foot down and said, we're not going to publish any papers unless you deposit the sequences that you're talking about in this GenBank database. And that was sort of the forcing function that caused kind of genetic data to start getting accumulated. And then there were a variety of different techniques invented for doing gene sequencing. I could tell you various side stories associated with that. But then there was kind of okay, let's see if we can mount an effort to do the whole human genome. And it was kind of, there were techniques for doing that, but one of the key techniques invented by Craig Venter was this idea of shotgun sequencing, where it's kind of like you break the DNA up into many pieces of length, I don't know, 30, 100, whatever else. And then you sequence those individual pieces, and then you've got this giant kind of jigsaw puzzle to assemble of seeing how all these pieces align with each other. And sort of the main insight there was you can kind of solve the problem with software, so to speak, rather than having to meticulously sort of work out how all the pieces go together in advance. You just sequence them all separately and then say, how do they fit together, by doing that as a kind of software algorithmic process. And that got figured out. So the human genome, when did it get? That must have been 2000, sometime in the aughts. The first sort of human genome got sequenced. A lot of it was Craig Venter's genome, his own genome, as it turned out. And I think some of the rest was a lab tech who had worked on the project's genome. But in any case, then it started to be possible to get genome sequenced. I know I got my genome, whole genome sequenced in 2010, which is pretty early. I think I was in the first few tens of people who got their genome sequenced because we happened to be working with the main company that was doing sequencing at the time. The idea of being able to sequence smaller parts of the genome, just an individual gene or individual SNPs, individual places where you can identify the particular base pair that occurs at that particular point, that came a bit earlier than that. And then, of course, there was sort of the whole question of, okay, if you've got your whole genome, what can you learn from it? And I know when I got my genome sequenced, I had somebody dig through it for a couple of months and just see what could you find. I was curious, and I remember the first email was, there are 700 and something diseases you might have, and it was easy to point out that most of those would have sort of killed me in the first week of life because and so we kind of know I don't have those particular ones. But it's been there was a lot of hope that sort of from the genome, one would be immediately able to figure out lots of kinds of characteristics and risks for different things, and questions of which drugs will be effective on a particular person and so on. And some of that has panned out, but it's been much muddier than one expected. I mean, I think it's still notable that you can't predict a human height from a genome, because probably height is something that is sort of a combination of the effects of a large number of different genes, and it's just still not known, even though there have been a lot of genome-wide association studies where you're trying to sort of use machine learning type techniques and statistical techniques to say sort of what features across this whole genome are relevant to this particular thing. That's been something that hasn't really been successfully figured out. And I think there was a wave of enthusiasm for sort of personal genomics that that has largely abated, and companies like 23andMe have fallen upon hard times. I mean, the applications that remain sort of out there are applications like ancestry determination, finding haplotypes. When you have a particular sequence, like on a mitochondrion, which is passed down the female line, the mitochondrion is a pretty short sequence. What is it? 10,000 base pairs, something like that. If you depending on where your ancestors came from, they will have slightly different genetic sequences, and you have to kind of characterize the genetic sequence by it. The whole thing is whatever it is, 10,000 bases or something like this, 10,000 letters. But you can kind of break that into certain haplogroups, and those are labeled usually with letters of the alphabet, and there'll be things like K2A or something as a haplogroup, and V1 something or other as a haplogroup, and these things characterize sort of the sequence on, for example, typically it's on the mitochondrion, which is passed down the female line, and the Y chromosome is passed down the male line, and that's been a sort of continuing, at least interesting application of these kinds of things. The other big application is in the case of cancer, where the genome of the tumor cells diverges at the beginning and increasingly, typically, as the tumor develops, and knowing what that genome is like and comparing it to the base genome of that a person has allows one to potentially target just the cancer and not other cells in the body. And so that's become an important and pretty standard piece of and knowing which chemotherapy agents are likely to be effective is something that is significantly determined by genomic features of tumor cells and how they compare to the base genomics of the patient. So those are two applications. There's some other kinds of things about, do you have risks for developing this kind of cancer? Do you have risks for this kind of disease or that kind of disease? That's been something that's been a little less effective. And certainly, there's a company that I think must have gone out of business because I stopped getting emails from them that would store one's genome, and then every time a paper got published that mentioned sort of different SNPs and genomes, you get an email saying, based on your genome and based on this paper that just appeared, you have a such and such percent probability of being left-handed, or a such and such percent probability of thinking that left-handed sugar tastes bitter, or something like this. And I think that, to me, somewhat segued into sort of more tarot card type soothsaying type activity, because the probabilities were just too muddy, and there just weren't that many things of the kind that Mendel had noticed of this color of pea is going to it's going to be definitively this, or human eye color, or human blood type, things like this. The cases where there are small number of genes and small number of SNPs that contribute, that's the exception rather than the rule. Gosh, let's see. Okay, so there are questions here about Des commenting, isn't some important part of how vaccines were discovered lost to history? I don't know. I mean, it's the kind of thing where it might have been known that things like this worked in ancient Chinese medicine, even in Western medicine, although I don't think in Western medicine. I mean, I don't think Galen and so on and Hippocrates mention sort of vaccine-like things in their kind of classical writings. So I think it was not known, at least in Western medicine. Let's see. Des is asking, when was the importance of epigenetics discovered or realized? So the question is, all the cells in our body have the same DNA, but yet it's clear that they have developed differently. Some of them are muscle cells, some of them are skin cells, some of them are brain cells, and so on. There's got to be something that goes beyond just the genetics of the cell that determines what type of cell it is. And also, it is the case that identical twins aren't develop things that are different. So there's got to be more to the story than just kind of the pure DNA. And I think the kind of the concept of epigenetics, the idea that there's sort of more to the story, I think that developed in the 1950s, maybe 1940s. So let's talk about kind of some more of this history. So it was sort of the picture of kind of how does life work in general. Owen Schrodinger, for example, had a famous book called What is Life that I think must be from around 1950, in which he talks about, actually, I think he talks about aperiodic crystals in there, by which, and I think that came before the discovery of the structure of DNA. So I think that was kind of a potential, how genetics might be encoded, I think was sort of a slightly physicist predicted kind of thing. I mean, meanwhile, there have been more abstract treatments, like John von Neumann had his whole theory of self-reproducing automata. That was also from the late 1940s, kind of this thing which ended up being a two-dimensional cellular automaton. The whole construction was finished by a chap called Arthur Burks. But the idea was, what would be sort of an idealized abstract version of the operation of life? And sort of the idea was, well, it's all this machinery, and it reproduces itself by having something which is essentially a program that describes what's going to happen, and then it's running the program to figure out what to do. And people have often said that was sort of a pre-version of what later became clear was really going on in life, which is, there's a program or a thing like a program that is what determines how the organism gets built. Now, the notion of sort of programs and tapes and ways to represent things, some of that is very old. Like, for example, Jacquard in 1800 had invented these punch cards for sort of specifying weaving patterns in looms. And that was something which, Charles Babbage made use of that in the 1830s, 1840s, in thinking about his analytical engine for doing computations, the mechanical engine for doing computations. That never got built. By the time Turing machines were coming around in 1936, Alan Turing was talking about a tape that would be kind of the way of representing what the Turing machine should do. And that idea that became very literal when the first actual computers were produced and one started actually using paper tape, which, by the way, had already been used in things like telex machines, just as a way to sort of pre-prepare the message and then run it over the telephone line once you were ready to actually communicate it. But then the idea of tape, and I realized that the first computer I programmed in 1972 or so was still programmed with paper tape. But this idea that there would be a tape that specifies the program, which is sort of a little bit morally similar to, oh, we've got this stretch of DNA, in humans about a meter long, a molecule that is this tape-like thing, is not quite so alien. The idea that there might be something like that would sort of specify the construction of an organism. But so that's the program. Then once the organism starts developing, well, then other things might be important. And one chap who was much involved with this was a person called Conrad Waddington in England, Scotland, I think, originally, who I never met. I met his daughter as a mathematician who I've known. He had the idea of what he called canalization. And that was a notion that you would have these sort of different things that could happen, and there would be these tracks that they would start going on that were tracks that were epigenetic. They were beyond purely the genome. Now that got all a bit mixed up with the idea of so-called developmental constraints. So when an organism, there's sort of this question in biological evolution of, can evolution lead to anything? Could evolution lead to animals with wheels? Could evolution lead to animals that have radio transmitters? Could evolution lead to whatever? And there's sort of some version of that was, well, evolution is so powerful, it can lead to anything. It was the idea that evolution is specifying which proteins you are made of and so on. That was a much later idea and much later clarification. But sort of the notion from Darwin's 1859 Origin of Species on was, at first, evolution is such a powerful force it can lead to anything. So then the question came up, well, why don't we see animals with wheels, for example? And that led to the idea of sort of developmental constraints. Well, even if you could have wheels, there's no way for them to grow that way. And also maybe there's no way for them to evolve that way, but that's a slightly different thing. So then there was this sort of mechanics of development mixed in with what's in principle sort of possible to specify in the genetic program. And that was certainly in the 1980s, when I was first working on theoretical biology kinds of things, this notion of developmental constraints was still very much a kind of, what's associated with how did the organism develop, and is the form of the organism, the fact that it has, antlers that curl up in this way or something like that, is that a consequence of the developmental constraints, the development features of the organism, or is that something that could have been specified any way you want by the genome? And that was somewhat confusing, actually, at the time. I certainly was myself much more concerned with kind of the mechanics of development with cellular automata and things like this than in kind of what the genomic programs would be. And just in this last year, I figured out a bunch about kind of how to think about sort of the selection of programs in a sort of idealized way. But at least back in the 80s, it was still very confusing. There were people sort of saying, it's all evolution, it's all natural selection, there's nothing else going on. And versus kind of there are developmental constraints, and it's kind of inevitable that you could get a stegosaurus with plates like that, but you couldn't get a this or that other thing. I myself was very much on the side of the fact that there was sort of a potential sort of theory of biology of things you could get and things you couldn't get. Now, it was somewhat mixed up at the time with kind of the trailing parts of kind of the religious debate about natural selection and so on, because the original kind of story had been, how are there are all these kinds of animals? Well, pick your religious tradition, but it's told in the book of Genesis or something for Judeo-Christian religion, other other religions, other places. It's sort of told how the snake managed to get to be the way it is, and so on. There's sort of a story to it, but fundamentally, it's that the critters of the earth were created by God in the way that they are, and that's all there is to it. Now, by the 1830s or so, people were discovering, dinosaur skeletons, and that was kind of there've been, other kinds of organisms that are now extinct or presumably extinct. I mean, pretty much the Earth had been explored by that time, so people, you had to be having stories about what happened at the center of the Earth and so on to have, live dinosaurs around at present time. But that was sort of the beginning of that, and then there were ideas about sort of how there were a lot of places where people were identifying kind of similar themes to lots of different kinds of animals. There was a lot of study of that. I think Charles Darwin's grandfather, Erasmus Darwin, was quite into that. There were quite a lot of books kind of explaining and showing the analogy between these different types of organisms and so on. And then trying to remember this, there was a chap called William Paley, who was much into this idea of natural theology. And I think he had a book called something like Wisdom of God Revealed in the what is it called? Pretty much in natural wonders, and kind of the idea that sort of what one is seeing in all the complicated forms of organisms is sort of a reflection of it's sort of it's vestiges of creation, I think was a term that was used. It's kind of the what we get to see of sort of the divine process of creating these organisms. So that was a thing in was kind of the prevailing assumption about how things worked. And then along came Charles Darwin, who'd sort of been on this voyage around the world on the HMS Beagle as a naturalist, and in particular, notably sort of noticed in the Galapagos Islands that there were these different separated islands and different species of finch that had developed beaks of different shapes and so on, which he sort of said, well, they developed those shapes because they were dealing with different kinds of trees and so on to get their food in different ways. And that was kind of a tip-off of this idea that these species might not be constant, but might be continually evolving based on natural selection, based on only organisms that were fitter to get their food from the tree or whatever would survive and go on. And you had to have this kind of idea that there was a natural variation between parents and children, but that then the children who were fitter for the environment would be more likely to have more children themselves, and on goes sort of the theory of natural selection. And there's a chap called Wallace who was operating in Asia, who also had sort of similar theories at the same time. And Darwin eventually kind of, after not having published his theory for a while, in 1859, he kind of sat down for a fairly short amount of time and wrote Origin of Species. But prior to that, he'd written books. I have his 2-volume work on barnacles. I guess he'd studied barnacles a lot, going around the world on a boat for a few years. It's kind of like he couldn't avoid kind of looking at the different kinds of barnacles that developed on the hull of the boat, and that was his 2-volume treatise on the Cirripedia was kind of his day job in a sense. Origin of Species was more like and this is something I've noticed, and I'm going to talk about it. Actually, his other day job was writing kind of travel books about what had happened on, as he visited these different parts of the world. They're rather nicely written. They have a lot of things that today will be seen as quite politically incorrect about observing the inhabitants of different places, and also about almost every kind of animal. He would describe things about the animal, and then he would say what it tasted like if you ate it, which at least for some modern sensibilities wouldn't really be the thing. But in any case, when Darwin had uh, put out his Origin of Species, that led to quite quickly to this whole, well, wait a minute, the Bible says it works this way. There's the sort of creationistic idea of how this works, and evolution says it works that way, and these things are sort of at odds with each other. And this kind of developed in it became sort of uh, a big debate and so on. A very notable moment in that was Thomas Huxley, who was kind of Darwin referred to as his bulldog. I think sometimes worried they didn't really understand the theory that well. But anyway, he was out bulldogging this theory. Thomas Huxley was very much a proponent of sort of scientific education for folks out in the world. I'm sure in modern times he'd be doing live streams, but in those days he was involved with what were they called? Well, some of the kind of efforts, particularly in England, to kind of develop. Oh my gosh, there was a phrase, and I'm trying to remember the whole thing, and it's awfully sort of a bit incorrect in modern times, but they were called working men's, not clubs, but something like that, which was a place where people would go and, like Huxley, would go and give lectures to the working people, so to speak, about science and such like, to kind of spread that knowledge. But anyway, Huxley became sort of the bulldog for Darwin, and things a bit came to a head. Bishop Wilberforce, is that right? I forget his name. Anyway, there was a notable bishop, I think he was the Bishop of Oxford, who took on debating with Huxley. And actually, there's a particular room in the Natural History Museum, what's now the Natural History Museum in Oxford, that I remember I once gave a talk at. And there's a plaque in that room that says, "This is the room where the big debate between this bishop and Huxley took place." And somehow in that debate, I don't know, it doesn't play well in modern times. It's much more Victorian. I mean, there's some exchange about how, Huxley was, the bishop had been expounding his various theories, and Huxley was like, "I would much prefer to be descended from an ape than to have anything to do with this chap over here," type thing. Um, and that somehow swayed the audience and became a kind of notable moment in that history. I mean, that history then sort of later kind of well, the thing to say about all of this was there was sort of a big push for evolution is science. There can be nothing but evolution that one has to talk about. And for example, in the US, the Scopes trial, I think that was 1920, is that right? Or 1920, sometime in the 1920s, that was a somewhat put up job of a school teacher who was teaching evolution, and that was in Tennessee, maybe. That was and the kind of the I don't think it was the ACLU, but it was some, gosh, I don't remember some national entity that was kind of promoting the idea of kind of uh, uh, liberal values or something, was kind of set up this kind of this moment of, would this person's teaching license be revoked, et cetera, et cetera, et cetera, or worse. And that was kind of that was the US version of this kind of the creationist thing is on the losing side, and the kind of evolution thing is on the winning side. But that turned into this really very strong thing by the 1970s and 1980s even, where it's like, if you believe in science, you believe in evolution. Anything other than evolution, you're not believing in science, which was actually very polarizing and rather wrong, because it was clear that there were all kinds of things that were relevant to the forms of organisms that we get that were not to do with kind of evolution can achieve everything. Every aspect of every organism is determined by some feature of its environment and its adaptation to the features of the environment. I mean, people like Stephen Jay Gould was one evolutionary biologist who I knew somewhat, who wrote a lot of kind of popular books about things which were sort of the features of organisms that were kind of just a consequence of sort of being sort of optimized for fitness versus things that were more constraints from development and other kinds of things. In more modern times, it got the sort of this must have been the 90s or even 2000s, this evo-devo, evolution versus development, kind of two sides of the same sort of story. But it was very, very polarized. I mean, even in the 80s, it was very polarized. People like Richard Dawkins were very much on the side of, everything that it comes from religion is all nonsense and evolution is everything. And there was sort of and everything, and sort of the only alternative to evolution is creationism, which just saying sort of the critters were put in were just put in the way they are in one fell swoop type thing. And sort of the idea that there could be a scientific intermediate ground wasn't really very well recognized. So the sort of story of epigenetics and people like Conrad Waddington and canalization and so on was sort of a bit of the intermediate ground, but it wasn't really talked about very much. Another person in this whole picture was Darcy Wentworth Thompson, who was this rather lovely Scottish gentleman who I wrote an elaborate piece about in 2017. 2017 was the 100th anniversary of a book he wrote called On Growth and Form. Darcy Wentworth Thompson was really a combination classical scholar and naturalist. I mean, he wrote books on things like Greek birds and Greek fishes. Those are books trying to connect sort of the fish mentioned in the Odyssey with the names of fish that we have today. It's an interesting problem to make that connection. But anyway, Darcy Thompson kind of sort of thought of himself as sort of continuing what Aristotle had started. Aristotle had sort of cataloged features of natural forms and so on, and he wanted to go on with that, sort of informed a bit by physics and by mathematics, and trying to sort of identify the physics and mathematics of the forms of growth and so on. Some of what he did, I mean, probably his most famous page of his books is a page where he shows the fact that if you stretch them, sort of all fish are really the same shape if you stretch them and distort them. A fish that's very long and pointy versus a fish that's very, very sort of tall and wide. If you just deform the shapes, you'll get from one fish to another. It's a little bit of a mathematically curious and unconvincing thing. I know from the biography of Darcy Thompson by one of his daughters that even before Growth and Form was written, he was at Christmas time, apparently, he would often draw a picture of a dog on a rubber sheet, and he would pull the rubber sheet in different directions and show that you could go from, the poodle to the St. Bernard to the Dachshund, etc., by just pulling the rubber sheet in different directions. And I think that became the inspiration for this this particular most famous page about fish shapes, which I don't think is mathematically terribly. It's a bit questionable. But in any case, Darcy Thompson had been a figure who really put sort of the geometry, the mathematics, the physics into the growth of things. That wasn't very well mixed with people like Conrad Waddington, who was more of a genetics biology type person, but it sort of began to mix. And then there was the question of, well, these different cell types, what were they? How were the different types of cells identified? So, for example, Stuart Kauffman, a guy who I've known for a long time, had a theory in the 1960s. He said, well, there's all of this kind of this logical network of different kinds of genes being switched on and off and other kinds of things in a cell. And there are different possible attractors. There are different stable configurations of what's being switched on and off that's sort of a self-reproduced, sort of self-amplifying or something that's self-consistent. And that there might be different forms of attractors for different types of cells. So that was a theory. When I first knew Stuart in the beginning of the 1980s, that theory was still very much alive and kicking as a possible theory of how different cell types might arise. Later on, more details about methylation and lots of sort of genetic modifications that can happen that don't affect the core sequence, but affect aspects of how it's transcribed in histones and all these different configurations of DNA and so on. That started to get unraveled. That wasn't unraveled until really very recently. And it's still not very, it's still not well understood. I mean, an example of a place where that gets used is in the Yamanaka factors, which are this way of going from a differentiated cell that might be something like a skin cell or something, and convincing it to kind of lose its differentiation and go back to being like a stem cell, a pluripotent stem cell from which all other cells can be derived. And that was a thing, what was that, 10 years ago? Am I losing track of time? 10 to 15 years ago, that was discovered that there were these, I think, 4 or 5 particular chemicals that you can use to make a cell revert to, more or less revert to its stem cell form. But that's a very recent thing. And there's still much more to discover about sort of how sort of the different cell types, the different kinds of markers on the surface of cells. These are all pretty active areas of research in biology and molecular biology. I mean, one of the things that doesn't help, which I guess I feel like I have a better understanding of now, is biology is sort of infinitely complicated. It's kind of a reflection, I think, of computational irreducibility and the fact that sort of biological evolution has just found these particular lumps of computational irreducibility that happen to do the things that lead to fit organisms. And as you study biology, you're studying the details of what happened to be the computational irreducibility that was found in this particular path of evolution. And it can be very, very complicated. And it can't be one of these things where it's often not one of these things where you can say, oh, I can remember that because it works this way and this way, because there isn't really a because. It's because it happens to just work. And that was discovered sometime, 300 million years ago by a critter very different from us. And that was sort of grandfathered through to the current state of life on Earth. But that's sort of another part of the story. Let me mention yet another part of this story, which is the kind of the competitor, in a sense, of Darwin in terms of evolution. In part was Lamarck and this idea of Lamarckian evolution, the idea that during the life of an organism, things happen to it, and those things that happen to it can be passed to its children. That it isn't purely a sort of random mutation, and you get in your life you get stuck with whatever you get stuck with, and then you pass that genetics to your children, but rather that the thing you pass to your children is changed by the experiences in your life. Now, clearly, when it comes to things we learn at a mental level, so to speak, then that is the case. I mean, if you learn this or that thing, you can pass it to your children, or at least try to see whether your children will listen to you if you tell them those things. But but there was sort of a theory that that things that would happen to an organism during its life would lead to this or that thing for its descendants. And famously in the Soviet Union, a chap called Lysenko was a big proponent of Lamarckianism, and I guess Stalin was sort of a fan of his, and that led to some pretty bad mistakes in terms of things about crops and crop breeding in the Soviet Union that came from that theory. And as a result, partly of that, Lamarckianism became sort of like creationism. Lamarckianism became sort of a thing, oh, we don't talk about that. That's a known to be wrong theory. Well, like so many things in biology, it's never as clear-cut as that. There are features of organisms that are passed on from one organism to another, not least because a fertilized egg cell has a lot of stuff in it beyond just the DNA. And so I think one famous example is people who were descendants of people involved in the potato famine in Ireland ended up with vitamin A deficiency, I think, which led to higher rates of color blindness in children, not because there was something genetic that had been passed down, but rather because of something that was, in a sense, epigenetic, that was a feature of, well, there's just not enough vitamin A, and that's not part of what is provided to the fetus, or possibly even in the egg cell, I don't know. And that was sort of a place where that gets passed down. But now there are endless examples of things like that, and it's so typical of biology that there's always another effect that that hadn't been taken into account. So as usual, it turns out that the idea it's pure evolution all the way down just isn't right. There's all kinds of issues from development and from the dynamics of biological evolution. The idea that there's only genetics passed down from an organism to its descendants, also not right. I mean, I think one of the features of Darwin and his belief about evolution was that he thought, now that we've got natural selection, we have a sort of precise theory that will explain inevitably the formation of higher animals and things like this, the increasing complexity of biology and so on. And he has, I think I've said this, it's one of my sort of favorite science history quotes, is the last sentence of Origin of Species. And I should actually learn it by heart, but I'm pretty bad at that. But basically it says, while the Earth is circling around the sun according to the fixed law of gravity, new species have been and are being produced, leading eventually to the production of the higher animals. So Darwin, my interpretation of that is that Darwin thought that just like there's a sort of physics theory, a mathematical physics theory of gravity that leads to the rotation, the Earth going around the sun, that there would similarly be a sort of precise kind of mathematical physicsy theory that would show one why the higher animals, the complexity of biology should increase that way. He never figured that out, and nor did anybody else subsequently. I think finally the things I was doing last year finally give one an actual way to see how that might work and to see the extent to which it is or is not inevitable that you get sort of this increasing complexity. And I think part of that has to do with the idea that you're just sort of sampling these pieces of computational irreducibility. Now, you can sample a complicated piece of computational irreducibility even pretty early in the history of biological evolution. And that's probably why the critters that you see in very ancient fossils are not really morphologically that much simpler than the critters that you see today. Now, of course, you're typically seeing in the fossil, you're seeing the outer boundary of the organism. You're not seeing, the details of the folds in this or that part of its brain or something like this. But anyway, that's sort of a story there. Let's see. Elsie asks, what have been my own interactions with early day or notable biotech people and companies and interplay with my own projects and technologies and developments and so on? Oh gosh. Well, I've sort of had a at a distance kind of interaction with lots of things in biology and life sciences for a long time. I mean, my own work, I've always kind of been a little bit afraid of biology because of this phenomenon that it's so different from physics and so on. And so any theory you have has exceptions type thing. But in the 1980s, for example, early 1980s, when I started working on sort of how do simple programs behave, one of my motivating features was understanding sort of the complexity of biological organisms. And so I worked quite a bit on that and got particularly into things like mollusk shell pigmentation patterns that are very complicated, but are rather easy to understand on the basis of these simple programs and so on. And I also, this was a time before the web when you couldn't kind of look up things about people very easily. And I think people knew that I'd been working on cellular automata. And so I got invited to all the theoretical biology conferences because people thought cellular automata, that has to be something about biology. Of course, it wasn't directly about biology, but anyway, so I got to know many of the people involved in theoretical biology at that time, and along with, well, people like Sidney Brenner, who had been, I guess I must have met Sidney after that time, but I would say he's, to me, one of the people who, like many of the early kind of molecular biologists, like Francis Crick and so on, had almost a very sort of proto-computational view of how things work. I mean, they were really interested in mechanism and in these kind of sort of how things, sort of explainable mechanisms for things. A lot of biology ends up being drowning in data and looking at a lot of very detailed features of, well, this has a slightly higher probability than that of happening, and it's really data, data, data. I think some of the early folks who were involved in sort of DNA and molecular biology and so on were very much more in the, there's going to be a mechanism. It's a physics-like thing. Now, sometimes that mechanism didn't pan out. Like there was a whole theory from physicists and mathematicians that it would turn out that the ways that you code for amino acids with 3 base pairs in DNA, that the code that was used will be an error-correcting code that will be sort of mathematically cool. Doesn't turn out to be true. It turns out the coding is much more just based on the chemistry and sort of what's a shape that's close to what and so on. But that was a little bit of a disappointment in terms of, we're going to go in, the physicists and mathematicians are going to go in, they're going to solve things in biology. But I think, for example, Sidney Brenner was certainly somebody who thought about things like Turing machines and their relationship to biology. Something that I learned as sort of a piece of trivia. It's kind of a funny connection and an indication, a correct indication of how small the world of foundational science tends to be. So Alan Turing died in 1954. His mother, Sarah Turing, wrote a biography of Alan Turing a few years later. And one of the things it was an obscure book, but back in probably around 1978, 1979, I got myself a copy of it, found it, and learned things about Alan Turing, and particularly Alan Turing's work on biological morphogenesis, which I didn't mention because it didn't go mainstream for many years, actually. But he, in 1952 and so on, worked on sort of a follow-up, in a sense, from the Darcy Thompson tradition of thinking about how you could use sort of physics and mathematics methods to think about kind of the forms of organisms. I mean, I think Alan Turing is the originator of the consider a spherical cow statement, and then he's looking at the modes of morphogen on the surface of the cow, leading to black and white dappling patterns and all that kind of thing. But anyway, back to Sarah Turing. So there's this thin book about Alan Turing, and I read this book, and I just remembered that, at the end, it felt a little bit like, Sarah Turing wasn't really that familiar with globalized science and so on. And she wrote at the end, how impressive it was that Alan Turing, when he'd done this work on morphogenesis, was getting requests for papers from places as far away as South Africa. Well, when I was a kid, this is now 1976 or something, so a full 20 years later, when I was a kid, I started publishing physics papers when I was like 15 years old or so. I started writing them when I was like 14 years old, and I would get these preprint request cards from all over the world. I could have made a stamp collection from them, from people requesting, copies of papers, because that was the way that one did these things before the web and all that kind of stuff. And so to me, it was unnotable that Alan Turing would have had a request from somebody in South Africa, because, I'd gotten them when I was a kid all the time. But so I sort of remembered that as a piece of kind of a reflection of Sarah Turing and also a reflection of Alan Turing. Okay, so I remembered that. And many years later, I was talking to Sidney Brenner, who grew up in South Africa, in fact, got his medical degree in South Africa and so on. And he was talking about how, yes, he'd been interested in Turing and Turing machines and so on. And he'd written to Alan Turing to ask for these papers and so on. So I then realized that was the connection, so to speak. So he'd been sufficiently interested, even when he was kind of studying medicine in South Africa and so on, to kind of sort of be thinking about these computational kinds of things. And that really went through, I would say, his whole life. I mean, he would come and see me every so often and say, you should be working on biology. And I'd say, I don't really know enough about biology. And he would say, but well, in my terms, he would say, but it's all computational in the end. I forget. I mean, he was always drawing these pictures of kind of sort of the mechanisms of biology and things. And in the way I would talk about it, it's kind of like these are computational mechanisms. So So yes, that's been one form of interaction. I mean, there've been many. I think I have to go soon here, but I noticed memes commenting that it doesn't know that Alan Turing was a progenitor of morphogenesis. What happened there was that the idea of reaction diffusion equations, the idea that there were chemicals that were determining sort of stripes and spots and things like this. Alan Turing called them morphogens. That idea was an idea that Alan Turing kind of initiated in his papers. Now, that idea has a complicated history because versions of that idea had appeared many times before, in particular in studying phylotaxis, which is something Alan Turing studied. Phylotaxis is the formation of plant organs like leaves and things, and it's the kind of the thing that determines sort of where the shoots come out in a growing stem, or how the pine cone pieces form, or how a daisy or something has little stamens inside the flower. Those are all features of this same phenomenon, which actually is that if you have this sort of growing plant shoot, the successive branches come out about 137 degrees away from each other, going up the shoot. That phenomenon has been rediscovered, probably 15 times. Its origin, its sort of mathematical type origin, which has to do with once a shoot comes out, it depletes the amount of plant hormone in that direction, and then you're kind of looking at where the next shoot is going to come out is where the plant hormone is going to be least depleted on the circle. So I discovered the explanation for that. People in the 1920s discovered the explanation for that. Alan Turing, I don't think he specifically talks about that in the papers he wrote, but he was sort of circling around the same kinds of issues. So it was sort of a known thing. But the question of what the morphogens were, what actually was the diffusing thing, that wasn't known to Alan Turing. I mean, later it turned out that things like retinoic acid were important. It became understood that things like the Hox genes were important, the homeoboxes were important in having something where you would have, it's either black or it's white, and it's discretely switching on or off depending on the concentration of this morphogen. And I think the one thing that was a little bit complicated is the whole theory of reaction diffusion equations. A chap called Ilya Prigogine, who became fairly famous in statistical mechanics and so on, kind of went off and sort of promoted this idea of reaction diffusion equations, and the idea that from reaction diffusion equations, you can get complicated patterns, even though the equations are somehow fairly simple to state. He really promoted that as sort of he called them, oh my gosh, what did he call them? It was a whole story of sort of out of equilibrium thermodynamics. And Prigogine even got a Nobel Prize, and although that was a complicated sort of story behind that. It was kind of, oh my gosh, what did he call them? Anyway, he had a name for them. Another chap called Herman Harkin had I think he called synergetics, which is sort of a related idea. But this notion of reaction diffusion equations, in my, I would say in my sort of, my impression from interacting with Prigogine and so on was that there was a certain degree of hiding of the Turing origin for this reaction diffusion idea. Because remember, Turing wasn't famous. In the 1970s, even going into the 1980s, Turing was not famous. And so the fact that he'd originated this thing wasn't a known thing, really. And so Prigogine really became the person sort of famous for the whole reaction diffusion equation and the Belousov-Zhabotinsky reaction, all this kind of thing, and wrote lots of books about this and such like. And the fact that subsequently these things got known as Turing patterns, but that was a subsequent thing. It may even be that things I wrote had something to do with that. I'm not sure. That's kind of historical shift. But yes, Alan Turing was very much in the middle of that and knew a bunch about it. I mean, he did that stuff in the beginning of the 1950s, and then died in 1954. So had he lived longer, he probably would have continued doing that. I mean, Alan Turing was a rather sequential kind of science researcher in the sense that, he did Turing machines, he did chess playing programs, he did code breaking, he did mathematical notation, he did, this, he did that, he did the other thing. And he really did them almost as separate projects, where kind of almost mysterious that when he came to study biology at the beginning of the 1950s, it doesn't seem to have occurred to him that he could use things like Turing machines to do that study. Rather, he was just sort of going back to the tried and true approach of differential equations that kind of one knew were the way that sort of chemistry and physics worked to try and study those kinds of things. All right, well, we ended up with just basically addressing just one question for all this time, but perhaps had some fun there talking about a variety of different biology, genetics, etc. topics. There's more to say, maybe another time. But thanks for joining me. And actually, I'm about to go to a day job live stream. So, for here, see you another time. Bye for now.